In this video, we're going to look at cell membranes. First thing is, what do you call a cell membrane? So, the cell membrane is obviously this layer around the outside of the cell. Now, when you're labeling it in a diagram, please call it a cell surface membrane or call it a plasma membrane. Sometimes it's been known that if you just write cell membrane, you don't get the mark because it's not detailed enough. So please call it a cell surface membrane or a plasma membrane. Now, first of all, the cell membrane is called a fluid mosaic, and it's called the fluid mosaic model. And the cell membrane is basically made of almost like a sea of these little phospholipids, which have got a little phosphate head with two little tails, okay? And then embedded in it are some proteins, right? Now, what can happen is that this is actually fluid because, excuse me, because these little phospholipids could actually swap places. Now, actually, some of the proteins can move as well. So it might be that protein at one time you take a photo, it might be over there, and then another time you take a photo, that protein might be over here. Okay, so the different parts in the phospholipid layer can move. All right, the phospholipids can move relative to one another, and the proteins, some of them can move, some of them are actually fixed. Now, we call that fluidity, and another way to kind of imagine it is imagine the phospholipid heads are like the balls that you get in a ball pit floating on a swimming pool. Imagine how they would stay in a layer, but they could swap places, couldn't they? They could move around within their layer. That's what it means by the fluid. The second part of the name of the model is mosaic, and that's just because it's made of proteins and phospholipids. It's also made of some other bits, cholesterol, etc. And they're all different shapes and sizes, like a mosaic picture. So the shapes and sizes, different shapes and sizes of the phospholipids and proteins, etc., make it look a bit like a mosaic. Okay, so we call it the fluid mosaic model, and they might ask you to explain what each part of the name means. So what does fluid mean? What does mosaic mean? If they just ask you in general, what does it mean? Make sure you explain fluid and you explain mosaic. All right. Now, the structure of the cell membrane, if we zoom into it a little bit more, is a phospholipid bilayer. The main part of it is made of phospholipids. So the phospholipid bilayer just means two layers, bi means two layers of phospholipids. So where on the previous slide I simplified it to one layer, it's actually two layers of phospholipids, two lines of them. Now, when you draw them, please try and draw the tails nearly touching in the middle. Don't draw them as little stumpy tails like that. I've seen that be rejected in some mark schemes in the past. So nice long tails nearly touching in the middle. We call that the phospholipid bilayer. Now, remember what a phospholipid is. You can see the detail a little bit more in the lipids video, but basically it's got a head and it's got a tail. And we need to know the properties of the head and the tail. So the head is what we call hydrophilic, and the tail is what we call hydrophobic. So hydrophilic things are attracted to water, and hydrophobic things repel water. Now, if we think about what this membrane is exposed to, the outside of the membrane is exposed to the cytoplasm, which is mainly water, isn't it? Cytoplasm. So outside the cell, outside here, all of this is mainly water. It's cytoplasm. It's going to be wet. Okay. Inside the cell, say down here, this is going to be Sorry, I said that the wrong way around, haven't I? So the inside of the cell, let's say this is the inside, it's the cytoplasm. The outside is what's something called tissue fluid. Now, if you watch the video about blood, tissue fluid, and lymph, you can learn more about what tissue fluid is. But it's basically what's around all the cells. So the inside of the cell is the cytoplasm, mostly water. The outside of the cell is tissue fluid, mostly water. It doesn't matter which side you put them on when we draw a straight line. And so these hydrophobic tails are nice and dry in the middle here, not touching any of the water. But the heads that liked the water, here and here, are touching the water. So that's why they orientate themselves that way round. Now, if you imagine another scenario where we've got, let's say, the cytoplasm water there and the tissue fluid water there, say we just had a single layer of phospholipids rather than a double layer. What would happen is as soon as this hydrophobic tail here touched the water, that molecule would spin around. It would touch the water and it would spin around because the tails would repel the water. And then what would happen is the tails here would touch the water and the molecule would spin around again. And so the molecule would just keep spinning round and round and round and round and round. So the tails will get wet, repel against the water, repel, spin around. Tails are wet, repel the water, spin around, and it wouldn't be stable at all. Whereas by forming this phospholipid bilayer, it's stable because the heads are hydrophilic and they're touching water and they're attracted to water, and that's fine. But the hydrophobic tails that repel the water are not touching the water. It's nice and dry in the middle here, and therefore that's why they form this kind of arrangement. So we've got our phospholipid bilayer that we need to know about. We know that either side is wet, the middle is dry, and that's why they form this shape. Let's have a look at what else we've got in our cell membrane. So we've got two types of protein. So we've got some types of protein that are just on one side of the phospholipid bilayer. And we've got another type of protein that goes all the way through the phospholipid bilayer. So you can see that first one on the left is just on one side, and you can see that big one on the right-hand side goes all the way through. So we've got two types of proteins that we need to be able to recognise and label. So let's just draw on all of those first of all. So this one here is just on one side, and the kind that are just on one side like that, we call those extrinsic proteins. The ones that go all the way through are called intrinsic proteins. So we've got extrinsic proteins and we've got intrinsic proteins. Extrinsic means they're just on one side. Intrinsic means they go all the way through. Now, sometimes you might see another name for them. The extrinsic ones are sometimes called peripheral proteins, and the intrinsic ones are sometimes called integral proteins. So just make sure that you're familiar with those words. That will really help. Let's have a look what else we've got in our membrane. So sometimes we have some little bits of carbohydrate. So here's my phospholipid bilayer. Sometimes we might have a little bit of carbohydrate, a carbohydrate chain attached to a lipid. So this red thing here is a phospholipid. This green thing up here is a carbohydrate chain. So together, that molecule would be called a glycolipid, because it's glyco means carbohydrate and then it's joined onto a lipid. Similarly to that, sometimes these carbohydrate chains can be found attached onto proteins. So I'll draw my protein there. So that big blob in amongst our phospholipid bilayer. You can be more careful with this than me. 2 tails per phospholipid head. So this big molecule here is a protein, and sometimes we get a carbohydrate chain attached to a protein, just the same as it attached onto that phospholipid, so that carbohydrate chain there. And this time that molecule is called a glycoprotein. So a glycolipid, carbohydrate stuck to lipid, glycoprotein, carbohydrate chain stuck to a protein. The final thing we've got in our phospholipid bilayer is cholesterol molecules. Now the cholesterol molecules just fit in here somewhere like this. They'll just look like a little shape in amongst there. And cholesterol is actually a type of lipid, cholesterol, and it helps to make the membrane fluid. We talked about fluidity at the start of this video. It just helps to allow the phospholipids to move around relative to one another and keeps the fluidity of the membrane. Okay. Let's now talk about the job of the rest of these parts of the membrane. So first of all, the phospholipids. So the role of the phospholipids is that they can control what enters and leaves the cell, which is what we describe as the function of the cell surface membrane. So they can control what enters and leaves the cell. And we'll look at how things enter and leave the cell in a moment. Actually, in another video, it's in the next video. The function of a intrinsic protein, the ones that go all the way through, is usually they are allowed large molecules to move through the membrane. So some look like this, and we call those channel proteins. And some of them look more like this, and we'll look at those again in the video that shows what crosses membranes. These are called carrier proteins. So those intrinsic proteins allow movement across the membrane, but movement of things that can't go through the phospholipids. The job of those little carbohydrate chains is for cell signaling or cell recognition. So what that means is that cells can recognise what type of cell another cell is. So, for example, your immune system needs to recognise which cells are yours and which ones are like invaders, non-self, foreign cells. So the way it tells which ones are yours is by those carbohydrate chains. Also, cell signalling, things like hormones can bind onto those carbohydrates to send messages to certain cells around the body. And then we've got the cholesterol, which we just talked about. And the job of the cholesterol is to keep the fluidity of the cell. All right, and that's the end of that video. We'll talk about how things cross the membrane in a separate video.